I'm Jay Phelan. Let's learn some biology. In the early 1900s, Gregor Mendel's work had just been rediscovered, and there was a lot of excitement trying to figure out how what we now knew about genetics could be integrated with what we knew about evolutionary biology. And one of the most pressing questions then, and it's a question that still confounds students today, was this: are recessive traits gradually disappearing from a population? If a trait's dominant, does that mean gradually it's increasing in frequency in a population? In particular, the most prominent geneticists of the day were curious. Is the trait of blue eyes a recessive trait? It's more complicated than a single gene, but is that trait slowly disappearing from the populations? And conversely, are brown eyes increasing because they are dominant? And a lot of it has to do just with the words they use. A dominant trait kind of sounds like it's taking over. Recessive, it's moving into the background. So it conveys this idea that there is a change that's going on just by virtue of being dominant or recessive. Now, there's a spoiler alert. They're not changing in frequency over time. Being dominant or recessive has nothing to do with whether or not you increase or decrease over time. But let's look at how they were able to figure it out. It goes by the name Hardy-Weinberg equilibrium. I'll get into that. But the way we approach the problem starts with just a basic identification of what we're going to be concerned with. Evolution we define as a change in allele frequencies in a population. So how would we know if that happened? Well, we'd have to know what the allele frequencies are, and then we'd have to monitor them again, and they'd have to be different. They'd have to change. Well, what is an allele frequency? Let's say, hypothetically, we're looking at one gene, and there are two alleles that you can have. Now, this is certainly not the most common or universal genetic system, but we'll use it because it's simple. We'll say big A and little a. Big A is a dominant allele, little a is a recessive allele, and the frequency of one of those alleles, big A, is just how many big A alleles are there in a population divided by the total number of alleles in the population. So there might be 5,000 individuals in the population. That means there's 10,000 alleles. There might be 2,000 copies of the big A allele. So when we want to calculate that, it's just 2,000 divided by 10,000, 0.2. So 20% of the alleles are the big A. That's the frequency of big A. And because frequencies total 100%, you have to be either one or the other. If we know that the frequency of the big A allele is 0.2, we know that the other one is just going to be 1 minus 0.2. So the little a allele frequency has to be 0.8. So that's it for the population. But that's how we talk about the alleles. Now, some of the terms that we will use that are just mathematical conventions people have come up with, when they refer to the frequency of big A, they call it P. When they refer to the frequency of little a, they call it Q. I don't know why, but that's what it is. If those are the only two alleles for that gene, P plus Q has to equal 1. There's nothing going on there other than saying they have to be 100%. But there's an insight that came from this. Once we understand that's how common one of them is, that's how common the other one is, it tells us something about how we expect them to come together in the population. And this is an important insight that these biologists had, Hardy in particular. He thought if the frequency of the big A allele in the population is P, well, how likely is it that an individual is going to have big A as their genotype? Well, what has to happen for that to occur? They have to inherit a big A from one of their parents. They have to inherit a big A from their other parent. We know what the probability is. It's P, the frequency, times P, the frequency. So the insight is that, if we have P and Q as the frequencies of big A and little a, then we know that the frequency of the genotype big A is just P squared. Similarly, we can say the frequency of little a, we have to get two copies. So it's going to be Q times Q, the probability of both of those events happening. So it's Q squared. Same thing goes for the heterozygote. How can you be a heterozygote? Well, you could get a big A from your mother and a little a from your father. What's the probability of that? P times Q. Or you could get a little a from your mother, big A from your father. It's an alternative way. What's the probability of that? Q times P. Two different ways, P times Q plus Q times P. It ends up being 2pq. That's going to be the frequency in the population of the heterozygotes. And this is an important take-home message. It's just straightforward indicating what everything is with the terminology. But if we know the frequency of the alleles in the population, we can predict which genotypes we should see in which frequencies in that population. A critical clarification here is that we're assuming that all the individuals are equally likely to mate with each other. So put simply, we call this random mating is occurring. That's, if you have these two probabilities, that's just one of the assumptions necessary for that math to work out. Let's work with an example. Let's say you've got a population of kangaroo rats. The kangaroo rats have 3 different coat colors. They have a dark brown, they have a spotted, they have a light brown. In a population, we could look at them and identify that out of 1,000 individuals that we had in the population, that there were 16 of the dark brown, which is the dominant homozygous trait, so big B, we'll call it. The spotted is a heterozygote. So in this case, we have a codominance or an incomplete dominance. The big B, little b, we see 222 of those. And the light brown, little b, we see 762 in the population. So if we have a population where we've observed those, first thing we'd want to know is, if we're interested in evolution, is what are the allele frequencies? Well, how do you figure out the allele frequencies if you've looked at these numbers of individuals in the population? It's very straightforward. To figure out the allele frequencies, you count them. We've got the dark brown kangaroo rat, 16 of them. Every one of those rats has 2 copies of big B. You got the heterozygote, 222 of those. Every one of those has 1 copy. And there were 1,000 individuals in the population, so that means there are 2,000 total alleles. So we have 2 times 16 plus 222 divided by 2,000, and that gives us our frequency of big B, which is 0.127. Now, how do we figure out the frequency of little b? Well, you could understand that together they have to equal 1, so you could just say 1 minus 0.127, or we could count them as well. We could say, all right, the light brown kangaroo rats, every one of those has 2 copies of little b, so 2 times 762. The heterozygotes, the spotted ones, everyone has 1, so it'd be 2 times 762 plus 222 divided by the 2,000 total alleles in the population, we get 0.873. So those are our allele frequencies in the population. And this was a big change from Mendelian genetics, because now we weren't really that interested in any one individual. What genes did they carry? We weren't really thinking about the gene as this sequence of DNA that was the instruction set for building a protein. Now, we're thinking about the population as a unit, like a cauldron. And in that, there's some number of alleles, and we're picking them out, and we're looking at how that changes over time. So we determined that P was equal to 0.127, Q is equal to 0.873. If we have these allele frequencies, we just go back to the insight that we had earlier. What genotypes do we expect to see? Well, we'd expect the big B to be the probability of getting a big B from one parent, probability of getting a big B from the other parent, which is just equal to 0.127 to 0.127. So that's our P squared again. And I'm showing a Punnett square here because that's a way of remembering what's the probability of this sperm fertilizing this egg, or this sperm could fertilize an egg with the other allele. So it's just a way of looking at all the possible outcomes that you could get when you have those allele frequencies in the population. So you look and you end up with 0.016, 0.222, 0.762. These are the genotype frequencies that we expect in that population. So if there were 1,000 kangaroo rats in this next generation, then we would expect 16 of the dark brown. We would expect 222 of the spotted. We'd expect 762 of the light brown. And for that to occur, we're assuming, given those frequencies of the alleles, that they're coming together at random. But we're also assuming that when they come together, that every one of those phenotypes has the same reproductive success. In other words, you're not more likely to get eaten if you are the dark brown or the light brown. It doesn't matter. It's irrelevant. So as long as they're all equally successful, as long as you're not adding new kangaroo rats to the population or taking them away. That's going to remove alleles from the population or add them, which would change the allele frequencies. So we're assuming that's not happening. We're assuming that no mutation is occurring, changing a big B into a little b or a little b into a big B or something else. As long as all those things are true, those assumptions that we're making, evolution's not occurring, and we still have this random mating, then when we count up the alleles in this next generation, we're going to find that they're still the same. You have some frequencies of alleles, they come together predictably, we calculate what they are, they're the same, as long as we're not changing them because we're seeing natural selection in the population or anything. And we say that the population's in Hardy-Weinberg equilibrium when the genotype frequencies stay the same generation after generation after generation. So you can think of it like Newton's first law of motion, that an object in motion is going to stay at the same speed unless a force acts on it. Or if it's not moving, it's not going to move unless some force acts on it. Hardy-Weinberg equilibrium says that as long as you don't have the force of evolution changing the allele frequencies, and as long as you have random mating, then they're going to keep coming together in the same frequencies generation after generation after generation. So we return to our initial question. Do the recessive traits gradually become less common? Do the dominant traits become more common? No. If there's no difference in the fitness of the organisms that carry them, in other words, it's not better to have brown eyes or better to have blue eyes or worse, then there's not going to be any change. They keep coming together in the same way. In the paper that was published that first clarified that the answer was no, they're not changing over time, the mathematician Hardy, who is a world-famous mathematician, he was even a little embarrassed to have written about this, but Punnett, the famous biologist who came up with the Punnett square, he asked him, they were friends, they played squash together, and he said, we can't figure this out. Does it increase? Does it decrease? Can you help me out? And apparently, Hardy, who was used to doing really complicated things, say, well, the math that's required is of the multiplicative table sort. And he said there's not the slightest foundation for the idea that a dominant character should show a tendency to spread over a whole population, or that a recessive should tend to die out. And he even wrote a massive autobiography and doesn't even mention this paper in it. He was kind of embarrassed by it. And I will also echo his feelings in that the math required is listed as the Common Core standards for 7th grade. So it's not a complicated calculation or computation. It's just an idea of how what we know about individuals carrying alleles can be viewed from a population perspective. So as long as we don't violate these assumptions, no mutation, no migration, no genetic drift, no natural selection, then we're going to see that the recessive alleles and the dominant alleles don't change over time. Now, if you're thinking about it, this might beg the question, well, those assumptions don't necessarily seem like they'd always be true, right? Natural selection occurs, mutations occur. So what good is a model when the assumptions that are necessary for it to help us out are likely not to be true? So, valid point, but it turns out still to be useful. You can think about in physics a frictionless surface or a perfect vacuum or light, the speed of light, where these are things we don't actually always look at or measure, but they're still useful as a system, as a model, as a endpoint. So we can still ask the question, let's say something like polydactyly. This is the case of having extra fingers or toes. It's a single gene trait, it's got 2 alleles. Do we expect populations to be in Hardy-Weinberg equilibrium for this gene? Well, you'd have to think about it. Is natural selection occurring on that? Does it decrease your likelihood of reproducing if you have extra digits? Well, you'd have to think about that. Maybe it does, maybe it doesn't. We're not really sure, but you can at least ponder it. If we're evaluating whether or not a population is in Hardy-Weinberg equilibrium, what we might find is that we expect certain genotype frequencies. We expect certain phenotype frequencies, But when we look at the population, instead of seeing the 16 dark brown kangaroo rats, the 222 spotted, the 762 light brown, if we see different numbers, maybe we see 100 more of the dark brown ones, 100 more of the light brown ones, and fewer of the heterozygotes, that's not what we expect given those allele frequencies. These numbers hold true for the same allele frequencies. What's going on there? Well, somehow they're not assorting the way we would expect them to. It doesn't mean that, all right, something's wrong, or we don't know, or our math is bad. What it tells us is that one of the violations of Hardy-Weinberg equilibrium has been violated. And that can be useful. That can tell us, in this population, evolution's occurring. And then we can look for it. What kind of evolution? Which ones are being more reproductively successful than others, and so on. Or maybe it's non-random mating is occurring. With those numbers I just gave, it almost looks like you're not getting very many of the heterozygotes. So maybe the dark brown ones are mating just with each other. The light brown ones are preferentially mating with each other. It might be something like that. So our lack of agreement with what we would have expected gives us some insight into what forces are acting on this population. That can be very useful. That can help us to understand how evolution works in natural populations. So anytime the phenotypic frequencies, the genotypic frequencies aren't what we expected, then we know that an assumption of the equations has been violated. And then we can, from there, move on to some additional exploration. In another video, I'm going to explore different populations and how we can analyze the numbers and what sorts of insights we can gain from them.